Hi, this is Mr. Wartsky again, and we are going to talk about cell signaling as a function or part of the cell membrane. We learned about the cell membrane in the last screencast. Now let's continue our discussion and add to our information. So we're going to start off with local signaling, and this is communication between one cell to another, and that's usually done through the gap junction in animal cells or plasmodesmata in plant cells. So in plasmodesmata, we have these little channels that go from one cell to the other through the cell wall here. Here's the channel. Cell wall is right here and right here. Well, in gap junctions, we have an open gap junction here, which acts as a simple tunnel so that ions and molecules can go from one cell to another. So that's local signaling. That's direct from one cell to the next. Now, at the same time, we could have a direct contact with surface receptors, right? So cells touch other receptors. And we're going to talk about this in the immune system because that's how some cells are activated by this direct touch between one cell receptor and the other cell receptor. Right? So we have cells touching each other or cells actually connected and transferring molecules back and forth. Now, local regulators, they are substances that are produced in one cell, they're released from the cell, they travel a short distance and bind to a nearby cell. So we have right here a diagram of one cell producing local receptors that are will bind and they'll bind to the receptors of other cells here. This right here is a neuron. This is, and the space in between is a synapse. And that's an example of a local regulator in which the neurotransmitters are dumped into the space here, which affect the next neuron. So we have papain signaling is when one cell produces a local regulator. So one cell produces, not once, one cell produces a local regulator. It leaves the cell by exocytosis and will bind to a neighboring cell, and it will bind to a receptor of a neighboring cell. Synaptic signaling, these only occur in neurons in which neurotransmitters move across the space. They're released into the space by exocytosis. They bind to receptors on the next neuron and channels open and you have a new impulse. And we'll talk about that later on in the semester when we talk about the nervous system. So we have short distance communication, in which one cell stimulates another cell in the local area. Then we have long distance. Now, long distance specifically deals with the endocrine system, and this is talk about endocrine signaling. In the endocrine system, we have tissues that produce hormones. Now, a hormone is produced by cells in one tissue. It's dumped into the blood, and it travels and binds to a cell of another tissue. So it travels throughout the body, and they'll bind to the receptors of cells somewhere else. Now, plants do have hormones, and we'll talk about it later in the semester, and they will be dumped into the vascular tissue, the xylem usually, and travel from one part of the cell to the other. There are two types of hormones. There's steroid and non-steroid hormones. We're going to talk about non-steroids first because they're a little more complicated than the steroid hormones. So in order to start our communication or discussion with hormones, we're going to know what a ligand is. And it is a molecule that binds to another molecule. And shape is extremely important. And you know that shape is everything. So if the ligand is the right shape, it'll bind to the right molecule, specifically receptors on the cell membrane. So as we talked about non-steroid hormones, we're going to focus our attention on receptors on the cell membrane. And there are two types. There are receptors that are bonded to the G protein, and there are gated ion channels. And I hope you're getting all this down. Remember, hit pause if you need to. So G protein. So the G protein coupled receptors, or the GPCR. The GPCR, or the G protein coupled receptor, is bonded to the G protein that's bonded to GTP. And the GTP will begin the reaction. So in our diagram down here, we have the GPCR. This is the receptor. And on the bottom, we have should be GTP here, right? And the GTP will start the reaction. Now the GTP starts a signal transduction pathway. Now the signal transduction pathway, don't get scared, is a multi-step process. It just amplifies the cell response, and you can happen in two ways. You can have phosphorylation or dephosphorylation, and removement of phosphates is really important. Or you can have a secondary messenger, and the secondary messenger starts a reaction, which eventually leads to the cell response. Protein kinases are what's really important in the phosphorylation-dephosphorylation reaction. They actually transfer phosphates from one molecule to another. When you transfer phosphates, you transfer potential energy, and you will change the shape of the molecule. Both things are very important. Kinases can cause phosphorylation cascade, which one molecule is phosphorylated, which causes a whole bunch of molecules to be phosphorylated. Protein phosphatases will remove phosphates from proteins, that's the dephosphorylation. It will inactivate protein kinases, and it will stop the signal transduction pathway. So phosphates start, dephosphorylation stops. Signal transduction, we see here, we have the binding here at the top over here. This is the molecule binds here. This is the G protein. It activates a molecule down here, which is a protein kinase, which activates another, right? Another protein kinase, which activates another protein kinase, and you go further on down, and they tell two friends, and they tell two friends, and they tell two friends, and pretty soon all heck is breaking loose, and the cell is reacting. Secondary messengers, there are two types of secondary messengers. We have CAMP, which stands for cyclic adenosine monophosphate, not triphosphate that we're used to, but monophosphate, and calcium. Calcium is one of those miracle minerals that is used by almost every system in the body. So CAMP, cyclic adenosine monophosphate, what happens here, this is an example of CAMP as a secondary messenger. Epinephrine, that thing that causes your heart to beat faster, fight or flight type response, binds to the cell receptor. The G protein will leave the GPCR, and G protein and GTP binds to adenyl cyclase, which is activated. This will break down many ATPs into many CAMPs. The CAMPs bind and activate protein kinase A, and that'll start the phosphorylation cascade. So CAMP is the secondary messenger because it does not bind directly to the molecule. The G protein receptor binds to the molecule, which activates the G protein, which will activate adenyl cyclase, which will break down ATPs into CAMPs or CAMPs, which will then activate protein kinase A. Kind of crazy if you think about it. Here's CAMP. Might be good to get this down, because I'm sure you'll see it later. Hit pause. Calcium. So calcium ligand binds to the cell receptor. The G protein will leave and bind to an enzyme called phospholipase C. I don't know what happened to A and B, but phospholipase C is the enzyme that we like. This enzyme will be activated and cleaves a special phospholipid, PIP2. And as PIP2 is broken down into diacylglycerol and IP3. No, it's not Italian Pizzeria 3. IP3 will bind to calcium channels on the endoplasmic reticulum. The ion channels will open and calcium flows into the cytoplasm, and the cell responds. So if you need, hit pause and get that down. But better yet, there's a diagram here that you can also take a look at. So IP3, yeah, see IP3 here, binds here, poop calcium out. Woo. Good job. Ligand-gated ion channels, we said that is another type of transport. So the surface transmembrane proteins will block the free flow of ions, can't get through the hydrophobic interior. When the ligand binds, the molecule binds, proteins change shape and the channels open. So the molecules can move. This is a crazy thing about all the different types of ligand paths, like the death factors at the bottom. Steroid hormones, a lot easier. They are lipid, they are hydrophobic, and they can pass through the cell and nuclear membranes. So the steroid binds to the protein in the nucleus. The new steroid/protein complex binds to the DNA, and that will increase protein production. So steroids can enter any cell, but unless the cell produces the protein to bind to the steroid, then the cell will not respond to the steroid. It has to have this protein inside the nucleus in order for the steroid to work. Here's a diagram of the steroids. See, we're binding to the protein here, binds to the DNA, increase protein synthesis, the cell responds. Might want to get that picture too. Good. So that is cell signaling in a nutshell. Hopefully you got that all. If not, you can either refer to your book, ask me tomorrow in class, or watch it over again. Have a good night.